going to talk about prokaryotes and viruses, the DNA that they have there, okay, they have inside. So prokaryotic cells they don't have a nucleus, right? So the DNA is going to be free in the cytoplasm, usually on a part that is going to be called nucleoid, remember? And the characteristics of this DNA is that it is circular. Remember that we said that the eukaryotic cells, they have two linear strands that they joined. So we will find that prokaryotic cells, they have circular DNA. But not just that. If you have a prokaryotic cell, such as like this, no, imagine the bacterial capsule, cell wall, everything. You are going to have this circular DNA, but also you are going to have like parts, little small strands of DNA all along the cytoplasm. These parts are going to be called plasmids. They are very important. Why? Because it's the way that bacteria can do like a pseudosexual reproduction, because they can exchange these little parts of DNA with other bacteria. So there is an exchange of genetic material. So they can have a little bit of more variability, no? Genetic variability. So this is the main thing of prokaryotic cells, and the other thing is the viruses, right? Viruses. Viruses, as you know, and they are so fashionable now, they are not real cells, but they do have genetic material, and they are, you know, parasites. That is clear. They are parasites, and they are very specific for the cell that they are infesting. So then, as we have plenty of different types of genetic material for each cell, you are going to have viruses that are going to have DNA, some are going to have RNA, some of them are going to have to be linear, some of them are going to be circular, some of them one strand, one strand. Other, they are going to have two strands, no, double strands. So many different ways of having genetic material to infest, basically. So these are the main characteristics of genetic material and the types that we can find. Now what we are going to study, if you go to continue to page 150, is the chemical composition of the DNA. Okay? So let's see. I hope you're understanding. If not, remember, after the video, you ask me through Edmodo any question that you have. Okay, so now the structure of DNA, or the chemical composition, okay, chemical composition of DNA is going to be what follows, right? DNA, what is it? It's a nucleic acid, right? And a nucleic acid is going to be made of little like pearls. Remember that we have like one strand, like one necklace, and this necklace is made of pearls. Each pearl is going to be one nucleotide, ¿no? Como un collar de perlas, ¿no? Cada perla es una parte del ácido nucleico y esa perlita que forma ese collar se llama nucleótido, ¿vale? Nucleotides. Okay, so we have DNA. DNA is a nucleic acid, and nucleic acids are going to be made by nucleotides. Many nucleotides joined together is going to be poly. Poly means in Greek, I think it is, a lot, right? Polynucleotides. Right? Understood more or less so far? Good. How is going to be the structure? The structure of a nucleotide is going to have three main components. One of them is going to be a sugar, right? Un azúcar, a monosaccharide. Remember that when we studied glucids, that the main principales monómeros of glucids were monosaccharides. So one monosaccharide is a 5-atom molecule that is going to be called, in the case of DNA, remember that here we are talking about DNA. It's going to be called deoxyribose. Yeah? So far so good. So this is a sugar. Sugar or monosaccharide, a glucid, and it has 5 atoms of carbon. ¿Vale? It's a cycle, it's un ciclo. We express it like this in chemistry. Apart from that, we have a phosphoric acid. And also, we are going to have a nitrogenous base. Base nitrogenada, ¿vale? Nitrogenous. Okay, so this is basically a nucleotide. Okay, just one. This structure is going to be repeated and they are going to be joined. Okay? First, telling you that we have different types of nitrogenous bases. You can see them on your book. We have adenine, we have thymine, we have guanine, and cytosine. Okay? That is when we are talking about DNA. We will see how it is when we talk about RNA. So basically, this is the structure of a nucleotide. But when I'm talking about DNA, I'm talking about the union of many nucleotides. So this one, that is just one, is going to join to another one that is going to be very similar. Why I say very similar? Because the main structure, we see this one and this one, is going to be exactly the same. The only thing that is going to change in nucleotides, in different nucleotides, is going to be the nitrogenous base. Because this one can be adenine, and in the following nucleotide, this can be guanine or so, right? So this is the part that is going to change in every nucleotide. So they have to join also. And how are they going to join? It's going to be called this bond I can't remember the name now. Phosphodiester, sorry. Phosphodiester bond. Okay? So the way that we are going to join this phosphoric acid with the next nucleotide, because they are going to join the phosphoric acid of one nucleotide with the deoxyribose of the next nucleotide. So you would have here the nitrogenous base and here another phosphoric acid. So this bond is going to be called phosphodiester bond. So when we join one nucleotide with the following phosphodiester bond, okay? And that is how we form nucleic acids, joining nucleotides between them. And the order of these nucleotides is going to be essential for the information that they are carrying, for the protein that is going to be synthesized. Okay, so this order is going to be called sequencia of nucleotides. Okay, so for now I think it is enough. Now I will tell you what to do afterwards with all this information. Okay, study. Bye bye.